Okay, let's pick up right where we left off. We were talking about inflammation. I said there's 4 steps to inflammation, and we'll talk about those separately in 4 panels at the bottom of the slide here. The first step is, of course, we're going to be injured. There's going to be some damage to tissue, and in this case, we've got a pin that breaks the barrier of the skin, and it causes damage to the tissue. This is going to cause the first step of the inflammatory response, which is chemotaxis. In chemotaxis, what happens is when the skin is damaged, the cells will release some chemicals, and they'll act as messengers or chemical signals. And these are going to be our histamines and our prostaglandins. And those are released by the injured mast cells, and that's going to cause a bunch of different reactions in the neighboring tissue. The second step that's going to happen as a result of those chemical signals is going to be vasodilation. So vasodilation, you have the blood vessels that are close to the site of injury will get bigger. So if you look at the size of the blood vessel in the first picture and then the second one, you'll notice that it's bigger. When those blood vessels vasodilate, they get bigger. That's going to allow extra blood flow to the site of injury. Also, what happens is the membranes lining the blood vessels change their permeability, and they allow things to move from the blood into the closest part of the tissue. Some of the things that are going to move in are platelets, tiny bits of the blood that are going to hopefully stop bleeding. It's going to start to form a blood clot that'll plug up this damaged part of tissue. And also we can have cells move out from the blood supply into the damaged tissue, which is going to bring us to step 3. Macrophage cells, yellow cells that you'll see here, those are going to move into the tissue because of the release of the cytokines in the first step in the chemotaxis. And those phagocytes are going to move in, and they're going to start to scoop up all of these bacteria that were introduced to the tissue. Okay, so they'll do what they do, which is detect stuff that isn't self and start taking it out of our tissues, breaking it down. Once they start to do that, once they start to clean up the area of foreign cells and foreign debris, those same macrophages are going to move into the last phase of inflammation, which is going to be tissue repair. So you'll see as we move from slide 3, from slide 2 to 3 to 4, that tissue, that wound, that damage is slowly started to be repaired as we get rid of all of the foreign particles and foreign bodies that moved inside of the cell, inside of the tissue, sorry, when the tissue was damaged. Now this slide is showing the exact same thing, a localized inflammatory response, and then the description down in the bottom gives you what's happening in the 4 different panels, and it's pretty much exactly what we talked about in the last slide. If you think about what we associate with inflammation, redness, pain, heat, swelling, it's all explained in these figures. We get our redness because the blood vessels dilate, and more blood moves to the area of damage, so it starts to turn our skin a darker red. When the blood vessel the cell walls or cell membranes change their permeability, we start to see fluid moving into the side of the tissue, which is going to cause our swelling. And of course there's going to be pain there because nerve endings are going to be stimulated by the prostaglandins, which is going to cause it a bit more pain, which pain is a message for you to try and protect that place so that that type of healing can happen and you won't further irritate any more infection. So that's the inflammatory response. Again, have a look at this video. It'll show you what we've been talking about the last few slides. And this is another summary of what we've been talking about. So you'll see the vasodilation, which is what we're calling the second step, because chemotaxis is the first step, increased permeability, and an inflammatory chemical signals. This is all lined out in this slide pretty well. a little bit more about the inflammatory response. The edema, the swelling, and I alluded to this a couple of seconds ago. When our blood vessels expand, they vasodilate, they're going to allow more things to leave the blood and move into the surrounding tissue. That's going to cause the volume of this whole part of the tissue to expand, and we're going to see swelling, which is the edema. The pain, as I mentioned before, is going to be turned up or increased in this area because of the release of the prostaglandins during chemotaxin, and it's going to cause the nerve endings to be a little bit more sensitive. It's going to cause a reaction to pain. But these two features of inflammation together, it's a signal to remind you that you should protect this area until the healing process is finished in order to release the chance of that infection or inflammation or inflammatory response moving to different parts of the body. Okay, so we talked about phagocytosis, we talked about inflammation, and now we'll talk about fever. We've all had a fever at one point or another. It's not terribly comfortable, but it is a very important part of fighting an infection. Now, a fever is an abnormally high increase in body temperature. Okay, so the temperature of our body goes up, and that's because we have cells that release chemicals called pyrogens. Now, pyro generally refers to fire, and gens in this case means to create. So pyrogenesis means to create fire. Well, these chemicals create not a fire, thankfully, but they do help reset the thermostat that we find in our hypothalamus. So that allows our body temperature to go up a few degrees higher than it normally would. The reason why this happens is this allows three things. It allows our body to speed up our metabolic reactions. Whenever you add heat to a chemical equation or to a chemical reaction, you're adding energy, and it allows that reaction to happen faster. So it speeds up our metabolism. It increases the effects of the antimicrobials, and we'll talk about that a little bit more at the end of this part of the unit. And it takes away nutrients from the microbes. So iron and zinc would be micromolecules that would help metabolism pathogens. At increased temperatures, they're not available for those pathogens, and so it makes their metabolism harder, so it'll slow down their growth. And that's what a fever aims to do. By increasing our body temperature, we do those three things. And a fever, of course, like I said, it's uncomfortable. It's not. It takes up a lot of our energy, which you always feel really tired, but it does a very effective job at fighting an infection. It's an innate response. It doesn't have any specificity, so you get a fever in response to any type of pathogenic or a pathogen invading or causing infection. All right, so the last part of our second line of defense are our antimicrobial chemicals. So in addition to inflammation and fever and macrophages, we have these antimicrobial chemicals that go around and add to our cellular defenses. Now, two parts of these antimicrobial chemicals are interferon, which is a response to a viral infection. So these are antiviral chemicals. And then we have a complement system, which essentially helps enhance phagocytosis. So we'll talk about those now for a little bit. All right, so here is the first one, and we'll talk about is interferon. So interferon is an antiviral protein, and it's released to interfere or to block viral replication. So we talked about how a virus replicates inside of a cell, and you guys know the different steps. So the idea behind interferon is it's released when cells are attacked by a virus, and it helps protect the other cells around it from becoming infected, or it helps to help those cells to stop viral replication. So if we have a look at the picture here, you'll see that a virus invades a cell and it goes through the regular steps. So we have uncoding, we have the viral RNA entering into the nucleus so it can be transcribed and then transcribed. But you can see here in their cell, once this starts to happen in the nucleus, we get these genes turned on for interferon. So this cell will release interferon, and that'll move into receptors for the second cell. So here's our first cell, and this one's going to be our second cell. Those interferon molecules will turn on genes inside the other cell, and it will produce proteins that will block the uncoding of the virus. So when the virus enters this cell here, cell number 2, it's protected against that viral replication cycle. So the virus can't replicate, it can't release new virons, it can't go on and infect any new cells. So that's the role of interferon. Here's a video you guys should check out, and again it's going to show you how interferon works to block viral replication. So that's interferon. The next set of antimicrobial chemicals that we have are our complement system. Now our complement system are made up around 20 big or large blood proteins, or proteins that live in the blood. They're produced in the liver, and they circulate in an inactive form. So they're not active all the time. They're only active when they're exposed to certain chemicals. So what they'll do is these complements will, one, they can enhance phagocytosis. 2, they can lyse or split apart bacterial cells or virus particles. And the third role that they play is they produce these fragments that can help regulate inflammatory responses. So these are 3 roles that the complement system have, and the reason that they call complements is because they go with our already with the methods that we have in our innate immune system. So lysing directly of cells, helping to increase phagocytosis, and helping with the inflammatory response. So this table right here is a really good summary of the roles that our complement system play in our immune system. So for all of these 4 panels, our complement proteins are going to be those little red triangles that you can see down here on the bottom, down here. And they'll show up in each one of these 4 panels. So one of the things that our component system can do is actual lyse bacterial cells, or target cells for that matter. So here we get our proteins. They find receptors on a pathogen or a cell that shouldn't be there, and they cause those membranes to bust, thus killing the cell. So that's lysis. Another thing that they can do is opsonization. Opsonization is a big word for helping phagocytosis. So in this case, we get our bacteria, and our bacteria are attacked by these complements. So these complement proteins, they come along, they surround the bacteria, and then they act as signals for macrophages. And the macrophages come along and they phagocytize and engulf these bacteria, and then they deal with them in that way. A third thing that we can have our complements responsible for is helping out the inflammatory response. So we can have complement proteins attaching to macrophages, and then those macrophages are carried to a site where inflammation is occurring, and they leak through the blood vessel into the tissue, and then they go out and help with our inflammatory response. Another role that we can see complements playing is the clearance of immune complexes. So this is something we'll talk about a little bit more in the next half of the unit. But we can see here our complements are attaching to antibodies, which have been used in a fight against an infection. And those antibodies are picked up by a macrophage or a phagocyte, and they're recycled so that we can use them again. And now this table here is a great summary of what we've been talking about today. These are all of our second line of defenses. We have our phagocytes, which ingest foreign particulate matter, stuff that shouldn't be there. We have inflammation, which will confine an infection and help fix it, tissue repair. Fever, which intensifies all of those reactions we just talked about. And finally, the antimicrobial chemicals, which are molecular defenses, the interferon, and the complement system.